Hello everybody, and thank you for joining us for tonight's update in partnership with St George Hospital. My name is Bertha and I'm the CPD and Events Manager at Central and Eastern Sydney PHN. Before we begin, I would like to start by acknowledging the traditional custodians and sovereign people of the land across which we work. I recognise a continuing connection to land, water and community, and pay respects to elders past, present and emerging. I'd also like to extend that respect to any Aboriginal colleagues who may be joining us tonight. Just a couple of housekeeping items. Please note that the session will be recorded and will be available on our YouTube channel within the next couple of days. At the end of the webinar, the evaluation survey will pop up on your screens. Please complete this to allow us to issue your certificates of attendance. To submit any questions throughout the evening, please use the Q&A box, which is located on the menu bar. Tonight's session has been accredited for one educational activity hour, and we will submit this on your behalf with the RACGP. I would now like to introduce our GP facilitator this evening, Dr. Martina Gleeson, who is a local GP and the clinical lead for South Eastern Sydney Health Pathways. Thank you, Martina.
Good evening, everyone. I'd like to start by introducing our speaker, Professor Emad El-Omar, who is the editor-in-chief of the journal Gut. His research interests include all aspects of the microbiome, inflammation-driven gastrointestinal cancer, and inflammatory bowel IBD. He's the director of the Uni of New South Wales Microbiome Research Centre at St George Hospital in Sydney, and the Professor of Medicine at the St George and Sutherland clinical campuses, Uni of New South Wales, and the director of UNSW Microbiome Research Centre at St George Hospital, Cogra. It's such an interesting emerging topic, and we look very much forward to hearing from you, Professor.
Well, thanks so much, Martina, and good evening, ladies and gentlemen. I'm speaking to you today from St. George Hospital. Let's just see if this works. Share. Can you confirm that you can see my slides?
Yes, I can, but yes, now you're in presenter mode. So
yeah,
that's better.
Thanks so much.
Mm-hmm.
So I'm the editor-in-chief of the journal Gut. I'm actually a gastroenterologist here at St. George Hospital and professor of medicine at the University of New South Wales. But I've been editor of this journal, Gut, for the last 16 years. And that's been a great privilege because it allowed me to oversee the developments in our field, and particularly in developments in the microbiome field, which is really quite dear to my heart. Our microbiome research center is here actually in your backyard. This is at St. George Hospital, and it was the result of collaborations between our local research foundation, the St. George and Sutherland Medical Research Foundation, with the university and the local health district, and it was officially launched and opened by the politicians at the time. This was in 2017. We have 5 themes that cover all aspects of human health, essentially: cancer, women's and children's health, infection, immunity, inflammation, mental health, neuroscience, and critical care, and more than 100 collaborative research projects across, certainly across Sydney and across the whole of Australia and internationally as well. So this microbiome, as you know, I think your patients will know quite a lot about it because they're very familiar with gut health. And almost every magazine or every newspaper or news article would have something about the microbiome. And rightly so, because this is something that is fundamental to our existence. You know, this extra organ that we carry, composed of trillions of microorganisms, mostly bacteria, but also viruses and archaea and fungi and so forth. And we are outnumbered by these cells. And these cells certainly have quite a profound metabolic output, equivalent to the liver, in fact. So as you can imagine, this would have a major impact on human health because it interacts at every level of the human body, and both in health and in disease. So the first take-home message is that this microbiome is a new frontier of medicine, and I think we're now advanced in this pathway, and we're ready for delivering those advances to human health. But we must do it right. We must do this in the correct way by providing the evidence base and explain how it's relevant to certain diseases and what we can do to manipulate it so that we can actually reap the benefits from this new knowledge. Perhaps this is the most important slide that I put up actually tonight, which is the concept that the human body does not work in compartments. So we can't be just cardiologists or gastroenterologists or, respiratory physicians. We have to be like in general practice. You're dealing with the whole patient. All these compartments are relevant to each other. And what happens in your gut will impact on other disciplines and other parts of your body, because there is no compartment in the human body that is sterile. We are exposed to microorganisms in every single compartment. There are no sacred compartments in the human body. And for that reason, I think this microbiome is very, very relevant to human health. This is an ancient concept. In fact, the Hippocrates, father of medicine, said all disease begins in the gut. And perhaps he wasn't referring to the gut microbiome, but certainly what we've learned in the last 20 or 30 years points to that basically being true. In other words, the origin of all of these kind of conditions may have something to do with the gut. Now, I guess the first question to ask is, where does this microbiome come from? And there's been a lot of work on this kind of epidemiology. And it's very clear that the babies, when they emerge, their first exposure obviously is to the mother's microbiome. So mom is responsible for the majority of that microbiome initially. So through the birth, whether it's normal delivery or through cesarean section, the baby gets mom's microbiota initially. And then there is kind of a specialization and kind of settling down of these microbiota and population of the certain sites within the human body. Now, the reason why that's important is because obviously moms are incredibly important for the health of the baby. And what mother passes on to her child is going to be relevant to the health of that child for the rest of its life. And as such, from a public health issue, and in fact, global health, there are many, many implications. So mom's health is absolutely very important for this. Now, beyond mom, then the baby obviously will mix with proximate individuals. See, these could be other family members, it could be the pets that are in the household, the actual inanimate objects, certainly the diet that the baby eats and so forth. All of these things will make up our microbiome, and then it takes up to 3 years, the first 3 years of life, for this to become steady and kind of at homeostasis, so that it doesn't have any major shifts or disruptions. Of course, there are different ways of interfering with the microbiome, and we'll talk about this towards the end of this lecture. Now, one very important concept about the gut microbiome is that of diversity. So diversity means different types of microorganisms that inhabit our gut. This is very important because that diversity equates with strength and resilience. So in other words, if we have lots of different types of bugs within our gut, that gives us resilience against infections. It allows us to withstand insults like antibiotics or different diets and so forth. So that diversity means skills, means these microorganisms have different skills that help the health of that individual. And that indeed is used as an index for a good or healthy microbiome. So if you analyze your gut microbiome, that would be one of the first kind of indices that we have, is how diverse is it? And that means how many different types of bugs with different types of functions. The other very important concept is that homeostasis. In other words, we've got really healthy microbiota, usually the probiotic type microbiota. These are the ones that impart a health benefit. And they're in kind of trying to fight off the bad guys, the pathogenic microbiota. And of course, if you have a healthy lifestyle, healthy diet, physical activity and exercise and so forth, you populate your gut with a probiotic type mixture and avoid this thing called dysbiosis. Dysbiosis is a kind of contentious term, but I think it just denotes the fact that the homeostasis is lost. So instead of having the healthy ones keeping the unhealthy ones under check, you have a little bit of a disruption and you turn this healthy microbiome towards a pathobiome, and that increases your risk of having disease. Now, how the hell does this collection, trillions of microorganisms, have such a profound impact on our health? Well, the answer is very easy. These are not inert companions. These are actually little factories that interact with whatever we provide them in the gut. So whatever you're delivering in the gut, be it diet, for example, indigestible fiber, plant-based components, and so forth, or whether it's medications or whether it's pollutants, all of these things, they will have an impact on the microbiota and will be metabolized by the microbiota. So they'll be broken down, they'll be fermented, they'll detoxify them, they'll toxify others, and so forth. And in the process, they produce chemical compounds, what we call metabolites. And these metabolites can then act on receptors, and those receptors will have an action. They will, initiate a response. That could be in relation to metabolism. It could be in relation to producing neurotransmitters or vitamins or activated compounds and so forth. And that's really the essence of this microbiome story. So basically, they're metabolizing whatever we deliver to them, and those metabolites then act on our receptors and have relevance to both health and disease. Now, the interesting thing about this is that we are all united in biochemistry, because that's fundamental to kind of science. So those metabolites will act on the same receptors in everyone, and our response will depend on what we're producing through those microbiota. So suffice to say that the gut microbiome over the last 20 years has emerged as a key kind of determinant of our health. It has a profound impact on our immune system, on our metabolism, cardiovascular, neoplastic pathways, and neurobehavioral kind of traits, and so forth. And that is why there's such a buzz about it, because it's relevant across all disciplines. Now, the most important thing that we need to remember is that it's manipulable. In other words, unlike our genome, with which we are stuck, we can actually change our microbiome. There are so many different ways of changing the microbiome, from one that is a pathobiome, in other words, nasty or bad, to something that is healthy. And I think this is the take-home message. Now, there are different ways of and different factors that impact on the microbiome. We already mentioned mom and the kind of the mode of delivery, so cesarean section versus vaginal delivery and so forth. That has an impact initially, at least. Of course, the diet and the mode of feeding, whether it's breastfeeding or it's formula feeding and so forth. And of course, the diet that we consume all the time. That's probably the most important determinant. But also other exposures like medications or, in fact, where you live, the geography, because that reflects essentially where you grow your food. That food itself in that geography would have a specific kind of microbiome that is also transferred to us. And of course, exposure to all the other factors that we mentioned, and also stress. So stress, both physical and psychological, will also have an impact on our gut microbiome. Now, host genetics, interestingly, you might expect host genetics to be relevant, but in fact, a lot of studies suggest that it's a minor role. Up to maybe 6% of the kind of composition and the functionality of the microbiome is determined by host genetic factors. And this is a good thing, because if your genes also determined what bugs are going to be growing in your body, we're essentially stuffed. I think it's good that we can actually change it over and above what our genes are. I think this is a very important concept. Now, so the take-home message here is that many factors impact on the composition and function of the microbiome, but it is manipulable. We can actually change it. Now, when you come to study the microbiome, and this is a little bit of kind of research philosophy here, the holy grail to be able to understand the relevance of something for the pathogenesis of a disease is what we call the longitudinal design. In other words, you want something whereby your starting point is before the condition develops. Ideally, you can collect all of your metadata, you can collect all of your biological samples and biomarkers and everything else, including your genome and metagenome and microbiome, before the condition. And then you wait for that condition to happen and then monitor what happens to all of these factors and then see the outcomes. In other words, were you healthy? Did you develop a disease or whatever? And then what happens to you when you get treated? Can we cure you, or can we just alleviate the symptoms and so forth? Now, that unfortunately is not feasible in the majority of cases, because if you're looking for, say, the development of bowel cancer, you've got to wait 30, 40 years before that happens. But there is one condition that is eminently suitable for this particular design, and that one is actually so predictable and so easy that we should really be using it as an exemplar of how to do these longitudinal studies. Of course, that condition is pregnancy. Pregnancy, thankfully, only lasts for 9 months. You definitely have a state when it's not there. You have it for a short period, which is 9 months, and then you can follow up all the outcomes. And this is why in our center here at the Microbiome Research Center, we decided to tackle this head on with perhaps one of the most difficult studies ever attempted. And that's called the mother's baby study. And that's essentially doing exactly what I mentioned, a longitudinal design that would start before pregnancy, preconception. We collect all the samples from mom and partner. And then we longitudinally follow up every 3 months, collect more samples and data and so forth. And then, of course, find the outcomes, whatever happens in pregnancy, whether it's a healthy one or a complicated one. And then we can predict then and study exactly what happens to all of these outcomes. I think this is a fantastic design. I'm really, really proud that we've managed to kind of initiate that study. We've recruited more than 600 women so far and with more than 300 babies born. So I think that really is a treasure of information that will be very useful in the future. This is longitudinal designs are very essential. The other big question, and this is relevant to all of the interpretation of the microbiome, and I'm sure all of you would be interested in this. In other words, when your patients bring in a microbiome report, what is it? How can we interpret this? So this is the definition of normal. What is a healthy microbiome? What is it that we aspire to have in our gut that could be representative of normal? Now, believe it or not, this is actually the most difficult question to answer because if you look at it from the perspective of the microbiology, in other words, what healthy bugs are there, invariably you'll find some, exceptions, like one bug that is healthy in one condition, maybe the opposite in another condition. So it's a little bit confusing. And to be able to do a multi-omic, very complicated kind of approach is obviously very time consuming and expensive. But if you're interested, this is an excellent review that we published recently in Gut that tells you what defines a healthy microbiome. This was published last year. Again, here at the Microbiome Research Center, we decided to tackle this, at least for our population, for Australia. And we started not from the microbiome end, but from the phenotype of health, like who are the healthiest people in our society. And we looked and we thought, well, elite athletes are obviously very healthy, so they would be interesting to look at. But elite athletes are very competitive, and maybe that competition is not a healthy kind of trait. But let's look at healthy people who are also elite athletes, but not competitive. In other words, they're not participating, they're doing it for fun. And that'd be interesting to look at. And then, of course, the ultimate health is in those who've survived all the tests and all the time. These are the centenarians that have made it to 100, yet retained, good mental function and so forth. And they're also be interesting to look at. So with all of these kind of phenotypes of health, we've managed to assemble what we think is the healthy, optimal Australian microbiome. And that's there is a core kind of signature of health that is defined amongst all of these healthy groups. And we will then use this as the basis for interpreting all the different disease signatures and figure out a way of how to get from one to the other. If you have a signature that predicts inflammatory bowel disease or, risk of cancer or risk of cardiovascular disease, then you can use that template to actually manipulate your microbiome back to this healthy, optimal Australian signature. Now, this will be very, very interesting, and I think this is a fast evolving field. And of course, it will rely on the use of very sophisticated, artificial intelligence to allow us to incorporate that kind of massive amount of data, and will be used to kind of dictate how we manage or manipulate this microbiome. Now, I'll just give you a few examples now of how the microbiome might be relevant to different human diseases. And I will start with something that is very common, which is cardiovascular disease and metabolic health. I think this is something that is obviously a major health burden, certainly here in Australia and across many, many countries. Now, suffice to say, this is a Dr. Stanley Hazen, who's the kind of the champion of the microbiome and metabolic health and cardiovascular health. He's from the United States and essentially has published many, many excellent original papers. And this is a beautiful review if you're interested in this topic, and showing very clearly how several metabolites that are produced by these microorganisms, usually in response to dietary components, so this dietary metabolism, have been linked to pathologies such as atherosclerosis, hypertension, heart failure, chronic kidney disease, obesity, and type 2 diabetes. So all of these kind of non-communicable diseases have a basis that, in this gut microbiome that contributes to it. Now, this is not just, fantasy kind of hypotheses. There's been a lot of amazing studies published. And again, this is another review by the same authors that summarizes it and essentially just gives you a kind of pathway as to how, eating, for example, the unhealthy diet that is, say, rich in red meat and fatty foods and so forth, and the production through these microorganisms of metabolites that have then a direct impact on the pathogenesis of all these cardiovascular and renal outcomes. The example is red meat, the choline and L-carnitine, and then the production of trimethylamine, and this gets by the gut microbiota, and it gets converted in the liver to trimethylamine oxide. And this is a very nasty kind of metabolite because it's been implicated in many of the pathophysiologic problems with heart disease, with hypertension, atherosclerosis, as well as renal failure and so forth. So beautiful kind of mechanisms identified. And if you are interested, I think this would be an excellent reference to seek out. Suffice to say, this is not just, again, just an academic exercise. These are studies that have shown that if you use your TMAO levels, plasma levels, this, look at the 5-year risk of death. This is in patients with heart failure. It actually outperforms all the other parameters that we know. So you can actually predict the risk of death by just measuring the plasma levels of TMAO. So I think all of these things are coming online very rapidly. We'll move on now to something that is more akin to my specialty, inflammatory bowel disease. Now, as you would imagine, this is, so the bowel, obviously the custodian of the bulk of the microbiology and the bulk of the microbiome in the body, will be obviously impacted quite heavily by the gut microbiome. Now, it's quite fascinating because you might think that is the trigger for inflammatory bowel disease. But that's far from clear. What we know is that obviously you've got a lot of risk factors. Some of them are genetic, that predispose to an abnormal response to the microbiology in the gut. So you might have something that, you overreact in terms of your immune system, your immune response to something that perhaps should be a commensal but has changed and become a pathobiont and so forth. But also the concept of the gut barrier. So if the gut barrier is leaky, and then there is this exposure through this leakiness, the immune system sees this and then mounts this kind of sustainable immune response, inflammatory response that is destructive and so forth. And this potentiates this phenotype. My own personal feeling is that obviously the microbiome is relevant, and there have been studies to show that if you use FMT, fecal microbiota transplants, you can actually restore that normal biosis and so forth. But I still think that there's a lot of research needing done to figure out what the initial insult is, that initial damage, to allow the microbiome to become relevant. So you've got the genes, you've got the microbiome, you've got so many other factors like your diet and your processed diet with all of its emulsifiers and other kind of additives and so forth. These are all relevant. But I still think that there is some factor that we haven't kind of identified that perhaps triggers that process. So that week, that particular period of time, you present with these symptoms. We still don't know why that happens at specific times. And I think that still requires a lot of research. Now, perhaps the most fascinating of all of the connections is the gut-brain connection with the microbiome. I think this is something that where probably all of the major advances over the next 5 or 10 years will be. It's not surprising at all that that there is a connection between the gut and the brain through the microbiome, because the gut is the largest nervous system outside of the brain. It's the largest endocrine organ. It's the largest immune organ, and it's basically wired up to the brain through the vagus nerve. And the enteric nervous system, a most sophisticated nervous system, is just sensing everything that is happening. Now, the gut microbiota, of course, are producing a lot of relevant metabolites. Some of them are neurotransmitters. Some of them are destroyers of neurotransmitters. So this kind of bidirectional interaction between the brain and the gut is very heavily influenced by the presence of these microbes and what they produce, those metabolites. And you're all aware of the fact that if you have some gut upset, you'll be feeling really miserable. So your cognition is affected. you're anxious, you might be depressed, your mental health is not right, and the other way around. I think it's a bi-directional kind of interaction. And it's not a surprise at all that there have been many studies now showing a direct link between the composition and the function of the microbiome and neurological disorders. For example, Parkinson's disease, multiple sclerosis, but also things like dementia, both Alzheimer's and otherwise. So this is a very, very exciting field. And it's exciting because potentially, again, you can intervene. You can change that gut microbiome and look for any changes in mental health or in neurological disorders. So beautiful studies. Again, if you're interested, there are some amazing reviews that are published that summarize that data. This is by Dinan and Crayon. This is from the team from Cork in Ireland. So world champions in this particular field. Now, one of the kind of the superstars of this thing is serotonin, of course. It's the critical regulator of the gut-brain microbiome axis. And it's that kind of, serotonin, the feel-good hormone that we're all very familiar with. Now, moving on to the microbiome and cancer, because that's probably something that we have already witnessed, not just a better understanding of the cancers, but also therapeutically as well. And I'll give you some really, really very interesting results. Now, as a gastroenterologist, we have the kind of a huge burden with cancer. The GI tract and its affiliated organs account for a very large chunk of human cancer, whether it's esophageal, stomach, or colorectal, pancreas, of course, and liver and so forth. All of these have a connection with the microbiology, and that's pretty evident. I mean, you're all familiar with, for example, stomach cancer and Helicobacter pylori. And stomach cancer and Helicobacter pylori is a very beautiful example of how a chronic infection in the stomach leads to the destruction of that lining of the stomach and the onset of what we call gastric atrophy, which fundamentally has the loss of physiology. So you lose the production of acid. So stomach cancer is a no acid disease. The acid goes out decades before the cancer develops. Now, why is that relevant? It's relevant because if you lose the acid in your stomach, you're losing your first line of defense against ingested microbes. So whatever you ingest, whether it's in your diet or if you just lick your fingers, you're having, touched something that is dirty or whatever it is, you rely on the acid in your stomach to sterilize whatever you've ingested. Now, if that is lost because of the destruction of the parietal cells by the H. pylori chronic inflammation, then that's why ultimately, decades later, you develop stomach cancer. So a very well understood model. Colorectal cancer is emerging at exactly the same, perhaps not to do with acid, but to do with the microbiome. So there is chronic inflammation that is manifest in those polyps, the precancerous lesions, and then as that inflammation progresses, you get colonized more and more by more aggressive microbiota like the fusobacteria. And interestingly, these are mostly actually oral microbiota. So things that live in your oral cavity that escape the stomach acid and end up causing problems further down in the colon. And likewise, there have been some fantastic studies on the liver and the pancreas. Unfortunately, I won't have time to go through all of these, but this is a very strong link. Now, this is fine about understanding disease and perhaps using these signatures as predictors of risk. But more importantly, what about the treatment of cancer? And this is where I think oncology has been, to some extent, galvanized and transformed in recent years by this slide here, the publication in 2018 of 3 papers back to back in one of the top journals called Science. And this just showed that you can actually predict response to immunotherapy by looking at those signatures in the gut microbiome, in your stool microbiome, which is, it sounds like sci-fi, but in fact, it does have a, kind of a logical explanation through the immune system. So the cancers that I looked at that time was, the classic ones that respond to immunotherapy, like melanoma, malignant melanoma, renal cell cancer, and non-small cell lung cancer. So all 3 studies published in the same issue of Science showed that kind of connection with the gut microbiome. And since then, that story has been developing very rapidly. And now there are actual studies designed to look at the role of taking FMT, fecal microbiota transplants, from responders to immunotherapy to non-responders. And as I said, this is something that is developing very, very fast, and I think it will impact on the way that we treat cancer in the very near future. Now, we still have to do a lot of work. We still have to figure out which FMT we transplant. What is the essence of that FMT? Is it just a whole FMT, or is it, certain taxa, certain types of microbiota that could do? All that is obviously subject to very intensive research. Now, I want to move on now to something that is much more fundamental, and that's diet, because I think diet is the most predictable way of actually impacting on the gut microbiome and ultimately impacting on manipulation of the gut microbiome. Now, there have been over the last year, and I will show you some of the latest papers published that have looked at this in a very, very sophisticated way and a very comprehensive way. And all demonstrate the value of diet and the importance of diet in different kind of ways. Now, this one was published in Nature Microbiology this year, and this looked at extremes of diet. If you look at vegans, for example, compare them to vegetarians, and compare them to omnivores, ones that consume meat and vegetables. And they showed, this is a very, very large cohort, 21,000 plus individuals. I looked at those that exclude major groups like meat or dairy or fruits or vegetables and so forth, and then compared their microbiomes and showed very nicely that ultimately we shape our microbiome, and that shaping of the microbiome is in our own hands by what we eat. So those dietary choices showed that in fact, if you consume a particular diet, that will impact on the composition and the function of your microbiome, but also how that diet was produced. So they found that in fact there's a small percentage of that microbiome is determined by microbiota present in the food itself, up to 3%, for example. And that's a direct impact on agricultural and food production practices. So that's a kind of a societal approach to food production. Now, all of these are relevant because they provide an insight into what makes the healthiest diet. Suffice to say that the vegan diet is not necessarily the healthiest. And of course, the omnivore is perhaps not the healthiest if you exceed intake of meat or fatty foods or carbohydrates and so forth. The vegetarian plus maybe some degree of other animal-based diet like fish and seafood and so forth, it perhaps gives you the best diet. And the Mediterranean diet, generally speaking, has emerged as perhaps the best one. Now, this kind of revolves around this concept of the whole diet and the whole health. And you'll be familiar with that concept where we as humans live on this earth, in this world, and we have to coexist with our ecosystems, with other humans, with animals and so forth. And we have to integrate all of this to ensure optimal human health, because we are all exchanging things between us, the animals, and the environment. Very relevant kind of concept, and one that is espoused by the World Health Organizations and so forth. Another study, which was also fascinating, published last year, and this shows how we all respond differently to diet. And that's based on the composition of the gut microbiome. So there's this kind of interpersonal variation in that diet microbiome interaction. And it turns out that in fact, depending on what microbiota you have in your gut, you can either toxify certain products. Toxify, that means you produce things that are kind of more antibiotic effect, and that's not necessarily a good thing. It leads to a disruption of your microbiome and potentially loss of resilience, as opposed to actually detoxifying some of the toxic compounds. So it's actually working for you. It's actually trying to reduce the harm from some of those nasty things that you're consuming. And that leads to microbiome stability and, of course, resilience. So that's basically what this study has shown. And yet another study, and this again was published this year, looked at the idea of non-industrialized type diet. Now, you're all familiar with diet nowadays and how it's labeled as heavily processed with lots of additives like emulsifiers and artificial sweeteners and God knows what kind of additives to it. All of this corrupts the diet because it's also corrupting the microbiome. All of these artificial things that we add to process the diet, make it more presentable, lead to addictive dietary behavior, leads to a pro-inflammatory change in the microbiota, leads to dysbiosis. So it's actually impacting on the health of our microbiome and the health of our and the gut health and so forth. So this study looked at the possibility of testing a non-industrialized type diet. So this is something that dates back decades, using diet that doesn't have any processing whatsoever, nothing added to it that is artificial, and combining it with one of the so-called lost microbes. So with civilization and industrialization, we have lost some of those ancient microbiota that used to work for us. In other words, used to impart a healthy gut microbiome and so forth. So this study looked at combining this non-industrialized type diet with one of those lost microbes. It's one of those L-Reuteri type probiotics. Now, this is not something that we have now. It's something that that is one of those lost microbiota. And to cut a long story short, it actually reversed all of these kind of effects of industrialization in the microbiome, and obviously impacted positively on body weight, fasting glucose, fasting cholesterol, as well as inflammatory markers. So this is a strategy that could potentially be adopted in terms of changing diet on a kind of a population level. Yet another study, and I harp on about this because I think it's within our hands, it's within our reach to control what we eat, and I think it's something that that we should actually learn from. Now, this study came from Africa, from northern Tanzania in the Kilimanjaro region, and then looked at the so-called African heritage diet. So, as you can imagine, this is a plant-based diet that is natural, it's not processed, and so forth. And this was a clinical trial where they compared urban dwellers with those that live in rural areas. So the rural areas consume this healthy traditional African heritage diet, whereas the kind of city dwellers, if you like, consume the typical Western diet, which is rich in fat and carbohydrates and so forth. And then there's a switch. So if you start with one, you switch to the other and so forth. And as a fifth kind of control group, they also tested this traditional fermented beverage, which is called mbegi, which is essentially a traditional beer made of fermented bananas and millet. And essentially what they found is that this switch from a heritage style to a Western diet affected different metabolic pathways that are classically associated with non-communicable diseases like obesity, diabetes, heart disease, and so forth, and promoted a pro-inflammatory state, chronic inflammation being the root of all evil. Now, in contrast, if you switch from a Western style diet to a heritage style diet, or if you consume this fermented drink, that largely led to an anti-inflammatory effect. Now, these are excellent studies, again, demonstrating that kind of the role of diet and so forth. Now, I'd like to also talk about something else that we as doctors we have that kind of privilege of prescribing medications, and medications have a profound impact on the gut microbiome. And I'll give you perhaps one of my favorite papers of all time. This was a paper published in 2018 in Nature, one of the top journals. And this tested the effect of non-antibiotic medications. So it took 1,000 non-antibiotic medications. In other words, it's all the medications that we use to treat every other condition, other than antibiotics, okay? And then tested these on a panel of commensal microbiota. So these are healthy commensals that live in our gut, and tested it in vitro. So they, tested these non-antibiotics, whether they can kill those bacteria or not, and found that a quarter of those 1,000 non-antibiotic medications had a direct killing effect on a number of those commensals. And if you want to put them kind of in ranking order of the most damaging to the least damaging, the top three that emerged were the antipsychotics. And you'll all be familiar with the side effects of antipsychotics. They cause obesity, diabetes, heart disease, strokes, and so forth. That's what kills the patients. And the second was the proton pump inhibitors. These are the acid inhibitors. And finally, the morphine, very powerful kind of opioid type medications. These were the three largest kind of offenders in terms of killing off your healthy commensal microbiota. Very important study, which means that what medications we give is actually has a profound effect on our gut microbiome. Potentially could explain some of the side effects and some of the beneficial effects, and I'll show you some examples of that. Now, I'm a gastroenterologist. I spent my first few years of my early career looking at H. pylori and peptic ulcers and acid secretion. So I know a little bit about acid secretion, how important it is. Now, this is just a picture of Sir James Black. And Sir James Black was a Scottish physician scientist who discovered two very important drugs, the beta blockers and the H2 receptor antagonists. And he won the Nobel Prize for his discoveries. Now, ever since, we've been obviously trying to stop acid because that's relevant to peptic ulcer disease and also for, heartburn and reflux esophagitis and so forth. And over the last 30, 40 years or so, we've had a proliferation of these acid inhibitors. Initially started with the weaker ones like the H2 receptor blockers, and then we had the proton pump inhibitors, which are far more powerful. And indeed, the final kind of frontier now we have, I'm not sure if they're available in Australia. I think they're available in the US and started in Asia as well. These are potassium competitive acid blockers, the PCABs. These are even more powerful than the proton pump inhibitors. They wipe out your acid completely. So your stomach is essentially neutral. Now, why is that important? Just to entertain you a little bit, I mean, just go back in history, just to remind you that vertebrates dating back 350 million years, all vertebrates have had a stomach that produces acid, with the exception of only 3 animals. Every other vertebrate has a stomach that produces acid. And these 3 animals are very, very fascinating. They're all Australian animals, the duck-billed platypus and the echidnas. And these are what we call monotremes. These are egg-laying mammals, exclusively obviously found in Australia. And this sadly now extinct gastric brooding frog that has a stomach that produces acid, but when it incubates its eggs in its stomach, produces a prostaglandin, a PPI if you like, that switches off acid secretion, allows the eggs to hatch, and it vomits up the fully grown frogs, and then it goes back to acid secretion. So what does that tell us? It tells us that it's an ancient function. Yet over the last 30 years or so, up to 10-15% of humans are walking around, with no acid in their stomach because they're on PPIs or on powerful acid inhibitors. Now, this just can't be right. I think you're losing that first kind of line of defense, and I think this will have an impact. And you, as general practitioners, obviously see all of those patients, and so my plea is to kind of question whether these patients need to be on the PPIs. Quite often, it's started by someone and never stopped, and there is no indication whatsoever for doing this. Now, going back to the effect of drugs on the microbiome, there are some medications that have a very, very positive effect on the microbiome, and one of them is metformin. And metformin has these, other than its role in diabetes and PCOS and other conditions, it turns out to actually have a profound effect on the microbiome, and it's a beneficial one. And there's been some amazing work. This was published a few years ago in Nature Medicine, showing what the mechanism is how it interacts with the microbiome imparts this healthy benefit. You'll be familiar with the GLP-1 drugs and so forth, anti-obesity medications, and every week now we get a report to say, wow, these drugs can protect you from renal failure, protect you from heart disease, protect you from not just diabetes, and it's not clear why they're so beneficial. And I think it's this is an invitation to figure out if they are doing something to the microbiome that perhaps impart a healthy benefit. And it's not a surprise because these GLP compounds are very important and interact very, very strongly with the gut microbiota and its metabolites. So there's that kind of bidirectional interaction as well. There's only been one study so far, and this is in an animal diabetic mouse model, that shows that if you use semaglutide, it does actually improve the gut microbiota, and this is relevant to that protection against fatty liver disease in that particular model of the diabetic mice. Now, we're doing studies at the moment to find out if these GLP-1 agonists and so forth are relevant to the gut microbiome and whether that explains their beneficial effect. Now, finally, how do we manipulate the gut microbiome? Well, there's a million things ways of doing it. Through diet, I mentioned exercise. Exercise also changes your microbiome towards a healthy one. Prebiotics, probiotics, all of these are potentially things that we can use. Symbiotics, that's the combination of both. Postbiotics, these are the metabolites from those probiotics that might be then druggable targets. Of course, some antibiotics and genetically modified microbiota and fecal microbiota transplants. So a very long list. We're blessed by having so many different ways of manipulating the gut microbiome. Diet, I've already mentioned, just this picture makes you feel very, very happy because it's very, diverse and beautiful and colorful. And this is all that correlates with beautiful health. So food as medicine is actually a very, very important strategy. And I think this would be, on a societal level and a political level, this should be actually the norm. Now, there are so many different other, ways of interfering. We mentioned all of these probiotics and so forth, and these live biotherapeutic products. Now, these are new kind of live microbiota that are able to do this probiotic function and so forth. If you're interested, this is an excellent review on one of the poster kind of child of the next generation probiotics. This is Akkermansia mucinifera. There's some very, very convincing evidence about its role. And then again, this review discusses the mechanisms and how it's relevant, healthy at least in quite a few conditions, but perhaps the opposite in others. So again, this Dr. Jekyll and Mr. Hyde is very important to take note of. It's not just probiotics that impart, a health benefit. There's a lot of ways of manipulating bacteria to actually impact on your human health. And there have been some absolutely fascinating studies published in the top journals in the last couple of years. This one is in Nature, and this was a probiotic that delivers a neoantigen to the tumor to encourage kind of a systemic anti-tumor immunity. And that's been, amazing using the kind of infrastructure of one of those typical probiotics, Escherichia coli in Nestle 1917. This is a kind of, again, a very, very famous probiotic type bacterium. So if you engineer it to produce this neoantigen, you could potentially use it to immunize against by presenting this neoantigen. And they've tested it in two models. One is colorectal cancer and one is malignant melanoma. So again, it's very promising. This is obviously still far from being used in humans. This is yet another manipulation of bacteria. This is Lactobacillus plantarum. And this was engineered to deliver something through the kind of nasal epithelium, the olfactory epithelium. So like you sniff this thing. This is done obviously in rats, but that nasal epithelium is very, very sensitive. It takes up whatever that bacteria develop and produces and delivers it to the brain. So you can potentially be treating brain disorders by having some, you potentially deliver it to the nose, inhalational, whatever, and that could actually provide a beneficial treatment. Yet again, this is another example of using these manipulated microbiota to withstand ultraviolet stress. This is all kind of fast developing models. Finally, FMT. Now, that's the quickest way of changing your microbiome. You just get somebody else's poo and that replaces the whole ecosystem. The problem there, of course, is that it's kind of potluck, what you get. So without actually having an intelligent approach of figuring out what is missing or what is wrong with your signature, with your microbiome, and who amongst all of us has the treatment, has the actual replenishment of that microbiome. That requires a very sophisticated and educated approach. And I think this is what we've coined as kind of intelligent FMT. That requires a lot of attention to that healthy signature and how relevant it is to the disease signature, and then finding the right donor to actually act as the one that provides that treatment for you. So these crab seals, so-called crab seals, I think will come on, but it requires a little bit of... Now, just finally, some guidelines in terms of the microbiome field. If you're interested, this is a very good review in Nature Medicine this year, clinical translation of the microbiome research. In other words, what does it all mean and how does, it's good background reading if you're, explaining to patients and so forth. Yet another one, the microbiome for clinicians, gives you some of the basic definitions and some of the basic kind of science, but in a very clinically friendly way. Again, an excellent review in Cell published this year. And finally, this kind of consensus statement that we published this year on international, this is an international consensus on microbiome testing in clinical practice, how relevant it is and what are the criteria that we should be using to impart that. Again, it's an excellent review, and I urge you to read that if you are interested. Finally, the future of microbiome medicine, it's all going to be artificial intelligence, because the amount of data that we're handling at the moment is massive, and that requires artificial intelligence, but this will allow us to personalize everything. There'll be personalization in clinical practice. This is coming. This will change the way that we do medicine. There'll be personalized diagnosis, treatment, prediction of side effects, prognosis, and so forth, prescribing personalized nutrition, personalized medications and therapeutics, and the use of all of these different ways of manipulating the microbiome, as I mentioned. This will all be coming online in the next few years. So we've got to be prepared for that. So final take-home message is that this microbiome is an exciting new frontier. It offers great opportunities for many disciplines. It needs to be studied properly and intelligently, and we need to provide that collaboration to work together to achieve it much, much faster way than we are doing at the moment. I think we're wasting a lot of resources by doing little studies here and there. I think we can combine the efforts and really move into translation and help our patients. And with that, I think I'll stop because I want to leave some time for questions if possible. I've got a few cases here, but we can discuss them, I think. Thank you so much.
Thank you very much. What an interesting discussion, although it just leaves the appetite for more. Someone's made a comment. It's interesting that IBD and HBP are associated with increased risk of Parkinson's disease. Do you have any comments on that?
Yeah. I mean, I think it's not surprising, I have to say. I mean, having been in this field for so long. But I suspect there'll be, again, some personalization of this one. So maybe certain people with IBD would have a certain signature that just happens to be the adverse one in terms of the risk of Parkinson's. I mean, the other thing that is fascinating is vagotomized patients. I mean, in my generation, the commonest condition that we dealt with as residents, as just junior doctors, was clerking patients for peptic ulcer surgery, because at that time H. pylori wasn't online. So a lot of patients had vagotomies and highly selective vagotomies, all that kind of stuff. So there is that cohort that looked at these patients, and then somehow they're protected, from Parkinson's. God knows, how the mechanism is, but now we know that things track up the vagus nerve, and perhaps if you happen to have the wrong metabolites and neurotransmitter kind of milieu, and that may be relevant to the pathogenesis of those neurological conditions.
Have there been any studies that show once a disease has developed that a gut microbiome, like I know we're talking about fecal transplants, but things like neurodegenerative disorders, a manipulation of the microbiome has been therapeutic for people with neurodegenerative disorders, or is that too late?
No, I'd like to think it's not too late. I think I wouldn't discount them at all, because there's certainly good experimental models for that, but no doubt about it at all. If you transplant young mice poo to older ones, you reverse some of those abnormalities, which is fascinating. There've also been case reports of patients with bipolar illness that have received an FMT, and there's a few celebrated cases actually that were in the news over the last 2 or 3 years, whereby bipolar 1 in particular, which is very hard to treat and not susceptible to placebo effects, but that particular patient was totally cured after. Wow. And in fact, that's the basis of, if anyone is interested, I think Professor Gordon Parker, who's one of the icons of psychiatry in Australia and the founder of the Black Dog Institute, has written a book on this, about those cases. And it's called a gut mood solution. So I think it's a fascinating link. But we're desperate to do the clinical trials. We would love to do those clinical trials with FMT in mental health issues and in neurological conditions. We have the capacity, the know-how to do these. All we need is the funding and the will from the government. But we're very happy to do those. Yeah.
Next question is, are you allowed to recommend what probiotics we should advise our patients to take? There are so many options, it's difficult to know what actually works.
Yeah. So I'd like to be a little bit controversial by saying I wouldn't advise any probiotics present over the shelf at the moment in supermarkets and so. It's very expensive, even if you source them from naturopaths and everything else. I mean, I have a respect for them. The concept is valid, but I'd much rather grow my own probiotics, to be quite honest, by adopting the measures that we've discussed. So if you go on a healthy diet and exercise, that will give you the probiotics that you need. And at this time, I think, as I said, we're working on next generation probiotics. Some of them are available. I'm sure Akkermansia is available, but I'd like to have a little bit more, clinical trial data for these so that we don't kind of fall into the same trap of jumping the gun and just adopting these things and selling them as food additives and food supplements as opposed to medication. So they don't have the same rigorous process to prove that they work. But in the interim period, just change your diet, and I think that is much more predictable way of manipulating your microbiome in a healthy direction.
And it's the same diet, isn't it? It like whatever we talk about, it's less processed food, make your shopping cart look like it came from a farm.
That's right. And
nothing in colorful cardboard packaging.
Absolutely. And also so plant-based components, if anyone is interested, so it looks like the more plant-based components, and the magic figure is 30 per week. Okay.
Oh,
It sounds like a lot. It's not actually. I mean, if you have a bunch of nuts, that's 5 or 6. If you have, fruit salad, that's another 5 or 6. It doesn't have to be industrial quantities, but it just means you've got diversity of sources of these plant-based foods, and they will all act to enhance your microbiome, even if your diet is probably otherwise imbalanced.
Not so good.
Just take these things, as a healthy kind of snack.
Someone has asked, would you recommend testing in GP? And if so, how do we do this? But what I'd like to add to that is sometimes a patient will come to me with the result of a microbiome test that their naturopath has ordered. I've got no idea.
Now, this is a very common scenario for me because they come to me and say, what do I
They
make?
know you're interested.
So look, what I'd like to see, and this is absolutely essential, is for us to establish this testing as part of routine clinical management. Okay, let's test it out. Let's call its bluff. Let's incorporate this into our clinic. Let's start with an example of, say, for cancer, the cancer care or whatever. You can start by collecting that microbiome signature before you do anything, and then follow the patients up. Whatever treatment you give, and see whether you could have predicted the outcome or the response to something. And then use that as the template for how you manipulate. And you just make it part of routine management so that ultimately we get there faster. The problem with testing, just generally testing, is that it produces exactly what you've described. A very unintelligible report that is fancy in terms of jargon and everything else. But ultimately, it's the same advice. Eat well, exercise, it's exactly the same thing. That's not particularly useful because most people that do these tests are the worried well. Yet I want to use it as the template for manipulation. I want to use it, for example, somebody presents with de novo IBD, and I test them at that stage, and I show there is a dysbiosis that could respond to this particular intervention. That would be the basis for how I manipulate it. That's a far more effective way to do it. And it's feasible. We just have to have the political will again to incorporate this knowledge into clinical practice.
Thank you. The questions are now coming in, so thank you for giving us time to ask them. If you take probiotics, and I think this is going to get back to diet, if you take probiotics, do you need to continue to take them to maintain the benefits, or does the gut take what you gave it and then populate the probiotics itself?
Sure. Look, I don't want to be the kind of spoiling the probiotic thing. They may work for you, they may not. The little data that we have suggests that very common scenario, and this was one of the cases that I had at the end, but no time to do it. Somebody takes antibiotics and say, well, obviously I've wiped out quite a large number of my own probiotics. Is it the right time to take probiotics? And they take some probiotics. That has been tested in studies, proper studies. And it shows that whatever you take, probiotics off the shelf, from supermarkets and stuff, may delay the recovery of your own microbiome after the antibiotics. So it may not necessarily be a good approach. That's not to say that some people will benefit, but it's just unpredictable. That's the problem. It's unpredictable. It's just potluck. So to a great extent, I think what I would do personally is if I take antibiotics, I would really make sure that I'm eating a very, very healthy diet, and at least as a kind of, an opportunity to repopulate with my own healthy probiotic that I had, microbiota that I had before.
Fermented food have a place to play in that?
Absolutely. So these are good.
Yeah.
These are good. Fermented foods are excellent because they contain natural probiotics. Not capsule probiotics. So these are good actually. There's good evidence to show that these fermented foods are excellent. Yeah.
Someone asked, is it possible for coronary artery disease to be reversed with a whole food plant-based diet?
Yeah, I mean, reverse is a very strong word, but certainly,
I know. Prevented
but
might be a
certainly
better one.
it will help. It will help.
Mm.
And the reason for that, I think, is because remember my slide when I said, there's no part of the body that is sacred that doesn't have any microbiome. Every time you brush your teeth, you'll be releasing some bacteria into your bloodstream. Now, if it just happens that you have a little plaque that is a bit unstable and, those few bacteria happen to be wandering past and so forth, that may be the trigger. I don't know. I mean, it's just, so that's just with brushing your teeth, let alone if you had a festering abscess in your oral cavity. And that's why periodontal disease and all of these things have been linked with cardiovascular disease, with dementia and all of these things. Now it's starting to make sense. But essentially, reversing, I think, is probably is a bit too strong. But certainly, you can prevent the worst of it. And certainly, if you adopt that healthy lifestyle that gives you a healthy gut, I think that will definitely protect you, and maybe protect you from getting more damage and exacerbation of that condition if you have a predisposition for it.
The questions are coming in thick and fast, and we're coming to the end, which is a real pity, but we'll try and forward some of them to you. I think I'm going to ask the one that is, what is the most important part of a healthy diet for a good gut microbiome? So in other words, what's the best prebiotic?
Yes,
So I
excellent.
think that's a good take-home message for us.
Yes, so grains, I think, so obviously fiber is the kind of because fiber will be metabolized in short chain fatty acids, which then give you a very strong immune system, a gut barrier function is excellent and so forth. So, so fiber is a key one. And from natural foods, so grains, obviously vegetables, fruit, nuts, seeds, legumes, all of these things actually are part of those 30 that I mentioned. That could make the best mix. And it doesn't have to be industrial quantities. And it's not per day, it's just per week. I think that would be probably the best mix to incorporate. And it's healthy and you feel so much better on it. I think it's remarkable.
I'm going to close it now because we've come to the end of our time, but thank you very much. And thank you to everybody who's asked questions. We will submit them to Professor El-Omar. Somebody also asked for a copy of your slides, but the recording of tonight's program will be up on the CESFAN website, so people can watch that video and grab those references as well. But if you could provide some good references for people to read, and we can send it back out for those who want to follow up on those studies, that would be really helpful. Thank you so much. This has been really, one of the things I became aware of as a medical student many years ago was that we were being told what was at the envelope. There wasn't more knowledge. This was what the world knew. And then as we continue, we get more and more knowledge, and so there's always more learning to be done. So thank you so much for your input tonight.
It's a pleasure. Thank you so much for having me.
Over to you, Bertha.
Thank you, Martina. Professor El-Omar, we just had one question which I thought might be relevant. Do you have any roles for GPs to be involved in your research?
Yes, absolutely. So when we were doing the mother's baby study, we were desperate for help, in terms of referring, potential, because we need them before they fall pregnant. In other words, they're thinking about how, so they will probably have seen their GP if they're planning pregnancy, and that would be very helpful.
You want to hope.
At some point, I will call upon you to help me out. But when we start recruitment again, I think at the moment we're taking a pause, but that would be definitely a study I need help with.
And just feeding back at the very end, there's lots of positive feedback in the chat. People saying, excellent talk, thank you so much. So that's nice real-time feedback for you. And we'll be sending out the feedback survey for people as soon as we click out of this, so that people can complete that for us.